Hey everyone, my name is Arman. I'm a second year master's student at McMaster University, and today I'll be discussing the work I've done so far in my master's, which has primarily been trying to contextualize antimicrobial resistance determinants by applying deep learning language models on biomedical literature. So, antimicrobial resistance is a growing problem in the world. Just in 2018, Canada saw at least 250,000 infections that resisted first-line antibiotics. Of these infections, 14,000 individuals lost their lives. Of these 14,000 deaths, 5,000 could have been prevented had they originally been susceptible to first-line antibiotic treatment. And the prevalence of resistance is growing still. And this could be attributed to two major factors. One, bacteria are really good at evolving resistance to avoid antibiotics, and two, they're even better at spreading resistance. As a result, resistance can spread from one environment on this chart called the confusogram to another, like from agriculture to livestock to humans and everywhere in between. To help control the spread of resistance, surveillance programs have been put in place to monitor resistance genes and mutations in priority environments like hospitals and farms. To track these genes, surveillance programs rely heavily on resistance databases like the Comprehensive Antibiotic Resistance Database here at my lab, or NCBI's Reference Gene Catalog. These databases primarily contain information about a resistance gene sequence, the mechanism of resistance, bacteria that contain the gene, and the drug classes it resists. However, they lack information about the sources these genes are found in, the epidemiology of these genes, information like the geographical regions, food sources, environments, and infection sites these genes are commonly associated with. This information is important for identifying priority genes and helping public health decision making. And this information is all contained within AMR publications. For example, if you were to read this abstract, they report finding NDM14 in Acinetobacter in a hospital in China. Right there we learn the bacteria, environment, and country a gene was found in. And there are thousands of papers like this published in the field of AMR, each one teaching us a little bit about a gene. By combining all this information together, we gain a big picture understanding of the genes that cause resistance. But as the number of publications increase year over year, it's important that we develop automatic methods of extracting this information. Thus, my project aims to do three main things: extract data, explore the data, and finally apply the knowledge that we gained. For one, I needed a reliable method of extracting this epidemiological information. This involves a combination of entity recognition to identify terms in biomedical text, followed by relationship extraction to identify related terms. With the data extracted, we could begin to analyze and explore trends in the data in order to summarize knowledge about these genes. And finally, we could also apply this knowledge towards predicting the transmission risk of a gene in different environments in order to calculate weights for these transmission events found in the confusogram. Extracting knowledge from a publication abstract requires two steps. First, we must perform entity recognition to identify all relevant terms in a text. For this work, I wanted to identify all AMR genes and epidemiology terms in the text. Here I highlight the relevant terms, color-coded by their category. So green is a gene name, orange bacteria taxonomy, brown an environment, pink geographical location. And the next step is relationship extraction, where we identify whether a relationship exists between two terms. For relationship extraction, we go sentence by sentence and see if a labeled epidemiology term within a sentence are related to the labeled gene term. In this case, we're looking at the first sentence here, so we take the terms and examine whether a relationship exists. In this sentence, they describe NDM14 being found in all these sources, so there exists a relationship between them all. This is simple to do for a single paper, but scaling this up to thousands of AMR papers requires an automated process. Thus, I trained some language models to do these two tasks accurately and reliably. The model I used is called BioBERT. It's a deep learning language model that's been pre-trained on biomedical literature, and since it's pre-trained, it only needs to be fine-tuned on a smaller dataset to complete the tasks we want it to do. So the authors of BioBERT compared its performance of entity recognition and relationship extraction to the current state-of-the-art models and found that BioBERT was able to match or even outperform the state-of-the-art models in both tasks. What this really means overall is that BioBERT can more accurately and reliably be used to do both entity recognition and relationship extraction. All BioBERT really needs is a sample set of data to learn from. And the pipeline for training BioBERT on these two tasks and applying the model is shown here and can be split into three sections. For one, we need to identify the relevant publications that potentially contain the information about genes we want to extract. I won't be going into this part in detail, but I created a classification model to predict whether a paper contains epidemiology information based on a given abstract. This gave us about 204,000 AMR papers to extract information from. With these papers in place, I took a subset of 10,000 papers from 2019 to generate data sets to train the BioBERT models. This data is used to train BioBERT to perform these tasks reliably and quickly on a larger set of papers. So starting with creating the data sets. The first data sets I'll be talking about is for entity recognition or identifying terms in an abstract that provide context. For this, I use different ontologies shown at the top that contain terms with relevant information organized in a hierarchical manner. So ARO, which was created by our lab, provides the resistance gene names, ENVO environments, food-on-food terms, GAS geographical terms, and so on. Using the terms and synonyms found in these ontologies, I used a pattern matching algorithm to directly match the term with the abstract text across 10,000 papers. From this, I got over 700,000 annotations across only 10,000 papers, and if you look at the numbers closely, there are a few oddities. For the geographical lexicon, there are over 300,000 annotations across only 10,000 papers. This would mean that each abstract mentions over 30 countries, which is impossible. When looking at the labels manually, the cause is really obvious. Many terms had synonyms that were short abbreviations that would match any regular term. For example, the geographical term American Samoa has the synonym as and mismatched over 10,000 times. To solve this issue, I filtered the labels through a series of steps. The graph on top shows the relative percent of annotations removed after each filtering step, and the bottom shows the relative percent of terms removed at each step. The filtering steps include filtering by length, stop words, nested terms, duplicates, and a couple final manual review steps. The most important steps based on their relative change were length filtering and the manual review steps. The manual review is especially important to generate high-quality data sets that we could really trust. This is important because the better quality of our data, the better the models perform after training. The final bar graph on the right is the percent remaining after all the filtering steps. And you can see that the geographical ontology in pink had the largest relative drop in annotations. To give some numbers, you could see the large drop in annotations before and after filtering, while the number of unique terms stayed relatively the same, only dropping about 2,000. This shows that a minority of terms caused the majority of mismatches. And here are the top 10 terms before and after filtering. You can see that before filtering there are many abbreviations adding noise to the datasets, and you can even see the American Samoa example at the bottom. After filtering, the annotations are all relevant terms like gene, E. coli, virulence, and hospital. With these datasets cleaned up, I moved on to creating the relationship extraction datasets. So now that we have datasets that can identify genes and epidemiology terms, we next needed datasets to identify if a relationship exists between these terms. For example, in the sentence, it is believed that this is the first report of colistin-resistant K. pneumoniae among swine strains associated with MCR1 plasmid in Malaysia, I would extract all the relevant labels based on the previous dataset and then say yes or no to whether the sentence describes an association between the two terms. Across 10,000 papers, there were about 12,000 potential relationships that needed labels. We've completed labeling the 12,000 relationships, but the data I'll be showing is when we only had about 7,000 relationships labeled. So I went ahead and fine-tuned the BioBERT models with the current datasets using different hyperparameters for learning rate and batch size. And from the training results for entity recognition, I selected the models that generalized the best. This meant that they were able to identify terms that belong to the same ontology without being shown those terms during training. Here I show the stats for the top performing models that were selected, highlighting the generalization performance in red. By plotting the number of unique terms in each ontology by the generalization performance, you can see that the more unique terms a lexicon has in their dataset, the better the model is able to generalize. This can potentially be because as the number of terms increases in a lexicon, the model is forced to generalize and identify patterns where a term may appear. But with lexicons with very few terms, the patterns are less obvious and the model learns that memorization is the best method to achieve the best performance. As a result, all models perform extremely well in memorization, but the three smallest lexicons, IDO, ENVO, and ESO, suffer with poor generalizability. This does not necessarily mean that these models are bad. Generalization really helps when the dataset is not fully complete with all the terms you wanted to identify. This is more important for large ontologies with many terms like NCBI taxon, GAS, and ARO, which have to deal with many synonyms, new terms being added, and misspellings in the text. The better a model can generalize, the better they can handle imperfect training datasets. But lexicons like IDO, ENVO, ESO, and FUDON don't have many terms to begin with, and these terms don't update as much. Thus, memorization alone works well for the most part. After training the models on the relationship datasets, I selected the best models based on the highest accuracy and F1 scores, followed by the lowest loss score. So the loss measures the amount of error between the predicted probability and the actual probability of the results. What is important to note is that there may be some bias in these models because of the imbalance of labels in these datasets. A label of 0 means that there was no relationship between two terms, and a label of 1 means that there was. In most of these sentences, there was a relationship between two terms, and this makes sense. In most cases, when something is reported in an abstract, it's a positive correlation between two things. Rarely did we find abstracts report negative results, like a gene was not found in X environment. And when there was a negative relationship, it was not because of an actual negative result, but because of a sentence would mention multiple terms, and only certain pairs of those terms had relationships while others did not. For example, in this sentence, MCR1 is related to E. coli but not Pseudomonas aeruginosa. This type of sentence accounted for most negative relationships. After training the models to identify terms and extract relationships between them, I applied the models to the remaining 200,000 papers that we had. This resulted in many annotations and many relationships. Here I'm just showing the predictions made for entity recognition models. A total of 2 million annotations were generated across 25,000 terms, and to properly interpret the results, I had to convert the predicted terms to a standardized set of terms. In this case, I used the original ontologies as my reference set. To do this, I mapped the predictions to the lexicons in two ways. First, I checked to see if there was a perfect match between the predicted term and the term or synonym found in an ontology. If there was no match, I calculated the Levenshtein distance between the predicted term and all terms found in the ontology. If they passed a breakpoint of 95%, I then assigned the predicted term to the ontology term. Levenshtein distance calculates the global distance between two terms and assigns a penalty for mismatched letters or gaps. One thing to note is that despite normalizing many terms, NCBI taxon and the GAS still had many non-normalized terms. Aside from some predicted terms not existing in the ontology at all, these two ontologies didn't do so well because of the underlying terms within the dataset. For the GAS, for example, the model made 80 predictions for the term Mississippi that could not be normalized. This is because the GAS only contains the term state of Mississippi, which falls below the 95% cutoff. To resolve this, I'll add a second layer of matching based on substrings, and the NCBI taxon lexicon didn't normalize too well because of abbreviations not existing in the original ontology. So when the model makes a prediction for E. coli, the term does not exist in the taxonomy. So what I'm planning to do is recreating the ontology with additional abbreviations to capture these terms. When looking at the relationships between these terms, you can see that the majority of relationships exist between normalized ARO terms and normalized epidemiology terms, shown on the far right. This means that despite there being many terms not properly normalized, it doesn't really impact the relationships as much, which is what we are focusing on. Looking specifically at the most common terms the entity recognition models identified, I plotted a box plot of the number of papers each term appears in each year, along with a heat map of the year those terms were found. For the ARO, the most common term is beta-lactamase, which is mentioned most often each year, but more recently MCR1 has been mentioned quite often as well, despite only having been discovered in the past decade. Looking at the environments, you can see that hospital is mentioned the most often by a disproportionate amount each year as well. From this, you get an understanding of the importance scientific research gives to each environment. You can also see that waste and wastewater treatment plants have only started to be investigated and thoroughly researched since the 2000s. For food items, the most common term is water, followed by common food products like cattle, poultry, pigs, and milk. And for geographical mentions, Japan and China are mentioned the most often. And here you can find the most common bacteria in literature, so E. coli, followed by Staph aureus and Pseudomonas aeruginosa. The most common anatomy parts are the blood, feces, and kidneys. Overall, these are the results of entity recognition, but after extracting the relationships between these entities and resistance genes, we can generate network diagrams like this. Overall, out of the 200,000 abstracts we annotated, 24,000 contained about 35,000 unique relationship pairs. The thickness of an edge represents the number of relationships between two terms, and the node size scales with the number of related terms. This is impossible to really understand on such a large scale, so by looking at these relationships on a gene-by-gene basis, you can learn a lot about a gene by itself. For example, these are all the relationships with the MCR1 gene, which were found in at least 12 abstracts. You can see strong relationships with E. coli and Salmonella, feces, urine, pigs, chicken, and many different countries as well. With this information, people who want to better understand a gene can quickly understand the commonly reported sources. Additionally, since these relationships have a set of publications they come from, you could learn more about a specific relationship by reading the associated literature. Not only can we use this information to understand relationships with genes and epidemiology sources, we could potentially use this data to understand how different epidemiology sources interact with one another. So coming back to this figure called the confusogram, it shows the different methods bacteria and consequently the genes that they contain can move from one source to another. And my goal is to see if we could better understand how likely transmission between these different environments are based on the relationships that I've gathered. So let's take a simplified example between food animals and humans. What if we want to know how common transmission is between these two environments? To calculate the risk of transmission between each of these environments, I took the approach of examining the genes that are shared between two groups. For example, between swine and hospitals, how many genes do these terms share a relationship with? If both swine and hospitals have a relationship with MCR1 and NDM1, then transmission potentially has taken place between these environments for those genes. Based on this overlap, we could create a weight between food animal subgroups and the term hospital, and by taking the average of these weights, we could also measure the weight between food animals themselves and hospitals. So far, I've tried two ways of calculating weight based on the gene overlap between environments. On the left is Jacquard similarity, calculated based on the overlap of genes divided by the total number of genes between these two epidemiology terms. And on the right is the numerator of Jacquard similarity, which is just the number of genes shared between these two sources. You can notice that in Jacquard similarity, there's a bright white spot indicating that these group of terms are highly similar. However, if you look at the right graph, it shows that those terms only share a single gene. And similarly, if you look at the right graph, there's a bright spot indicating a lot of overlap between genes, but their Jacquard similarity is not extremely strong. So neither metric works really well, and I'm currently working towards developing a better metric in the future. Overall, the work that I've shown here provides a platform to extract knowledge from publications to better understand resistance genes. Hopefully in the near future, this work will also reveal knowledge about transmission dynamics and the risk associated with different genes based on the environments they're found in. I would like to thank my supervisor, Dr. Andrew MacArthur, my committee members, my volunteers, my Google collaborator, and the MacArthur Lab for all their support. Thank you.